Hello and welcome to Science Research Weekly, Season 4, Episode 15. How about a cephalopod? Get ready for Rotating Universe. Universal Cephalopod PCR Primers. Moonglass solar panels. R's How About a 26? SAS For Dummies 3rd edition. And a grant for Life Sciences Entrepreneurs. This episode covers material from April 11th to the 17th. Let's get started with some research articles. First up, can rotation solve the Hubble puzzle? We've talked about the Hubble tension before, discrepancy between the value of the Hubble constant in observation versus theory, and it's getting worse. Well, this paper looks at how a slowly rotating universe might fix that issue. Next up, development of universal PCR primers for the environmental DNA metabarcoding of cephalopod mollusca diversity. Researchers use universal PCR primers for cephalopod amplicon sequencing of the 16S mitochondrial RNA to detect species of cephalopods like squids and octopuses via this eDNA extraction from marine water samples. Here, if you're looking at the video portion of this, is the graphical abstract. They're able to, for both octopodiforms, so octopuses, and then decapodiforms, so squid, they're able to get PCR primers. They can take samples from the ocean, get out environmental DNA, and actually barcode a variety of species all the way up to giant squid. So very interesting setup there. All right, moving along, we have acoustic modulation of mechanosensitive genes and adipocyte differentiation. Researchers were able to simulate cell lines with sound and found that they responded to it with differential gene expression. Never think about cells responding directly to sound, but apparently they do. And finally, for our articles today, we have moon photovoltaics utilizing lunar regolith and halide perovskites. Perovskites? I'm not sure how to pronounce that. I'm not a geologist. Anyways, researchers outlined a way to fabricate solar panels using regolith-based moonglass. So the idea here is that astronauts could establish a moon base and synthesize a lot of the heaviest portions of a solar panel there so they wouldn't have to transport it all the way from the Earth. So kind of interesting use of in situ development materials. All right, let's turn our attention now to some research tool. New articles dropped from the Journal of Statistical Software, Volume 112. There is parsimoniously fitting large multivariate random effects in GLMM-TMB. There is stability selection and consensus clustering R, the package Sharp. And there is true skill through time, reliable initial skill estimates and historical comparability with Julia, Python, and R. On the SAS frontier, Chris Hemedinger on the SAS Dummy announced the 3rd edition of SAS For Dummies, covering SAS 9.4 and SAS Via, and with chapters about things like SAS programming, data management, reporting, machine learning, and data visualization. So he was very excited to produce that. Apparently it sells well, even the older editions. So this new edition will bring SAS into the most current year. All right, on the R front, a new version, R 4.5.0, How About a 26, has been released, just so you can check that out. In fact, moving over to our bloggers post, the first 2 posts are on R 4.5.0. First, from Blog on Incredibly Curious, goes through it, getting excited about the penguins dataset now in base R, and also other interesting posts or points, including the issues about reinstalling our packages. Again, for minor version changes. Second, from the Jumping Rivers blog, more love of penguins. Things with parallel downloads, C32 and grevpv, all on R 4.5.0. Other R bloggers posts 3 and 4 rounded out here. There is the Jumping Rivers dashboard gallery. So looking at interesting collections from Jumping Rivers blog. And final is getting my feet wet with Plumber and JavaScript from R on Every Day is a School Day. Using Plumber to create an API for an interact and record event. Like, for example, monitoring a migrant event, hitting the button, and then pulling out something like barometric pressure. So very interesting sort of use case. I thought I'd be, up your alley. All right, let's wrap up this research tool with some 3 new CRAN packages. There is FastQRS, fast algorithms for quantile regression with selection. VAYR, extensions for ggplot2 to visualize as you randomize. And finally, E2Tree, explainable ensemble trees. For our close here, we actually do have some research funding to get to. Some new grant opportunities, first from the NSF, incorporating human behavior into epidemiological models, which supports research that incorporates social and behavioral processes in mathematical epidemiological models. And then second, from this thing called Bioinnovation Institute, it's the BioStudio program. It supports a world leading entrepreneurial PIs in translating innovative research ideas into new products and solutions for the benefit of people in society. So now the specific focus looks to be on brain health. And this institute is based in Europe, but the eligibility criteria says PIs can come from anywhere in the world, so it's not just limited to Europe. All right, that is our time together this week. Happy preemptive Easter, everyone. Thanks for joining me for Science Research Weekly, Science On.